All right, let's dive right in. Welcome to this rapid review of clinical oncology and tumor biology. We're going to run through a continuous, high-yield gauntlet of questions, so get ready to test your knowledge.
Starting us off, staging and surgical oncology.
Question 1. What biological event defines the transition to stage 4 systemic disease? Is it A, invasion of superficial basement membrane, B, presence of distant metastases or M1, C, sudden p53 mutation, or D, direct skin extension? Okay, the correct answer is B. So the absolute crucial point here is that in solid tumor staging, stage 4 is defined by M1, distant metastases. Once those malignant cells see distant organs, the disease is officially systemic. And this exam pearl is huge because it completely shifts our primary management goal away from local surgical cure, squarely into multimodality systemic palliation.
Moving to question 3. What distinguishes neoadjuvant from adjuvant therapy? A, neoadjuvant is for recurrence. B, neoadjuvant is prior to surgery, adjuvant is after. C, adjuvant uses vitamins. Or D, neoadjuvant is for benign lesions. The answer is B. Neoadjuvant therapy happens right prior to definitive surgery to shrink and downstage that primary tumor. Adjuvant therapy, on the other hand, comes after resection. Its specific job is to target and eliminate any occult, microscopic residual disease that might still be hiding in the body.
Question 4. What does an R1 resection surgical margin signify? A, no residual tumor, or R0. B, microscopic residual tumor at the margin. C, macroscopic gross residual tumor, or R2. Or D, benign tumor with no margins. The correct answer is B. Getting this precise surgical classification down is key. R0 means a complete, perfectly clean resection. R1 means that even though the surgeon couldn't see any tumor left behind with the naked eye, the pathologist found microscopic residual tumor cells right at that inked margin. And R2? That means macroscopic, gross tumor was visibly left behind in the operative bed.
Let's look at question 6. What is the most common site of distant hematogenous metastasis for extremity soft tissue sarcomas? A, liver, B, lungs, C, brain, or D, spleen? The answer is B, the lungs. You see, soft tissue sarcomas metastasize almost exclusively through the blood, rather than the lymphatics. And because the systemic venous drainage from your extremities flows straight back to the heart and immediately out to the lungs, that pulmonary capillary bed is literally the very first downstream trap those circulating tumor cells are going to encounter.
Question 7. What is the fundamental definition of a sentinel lymph node? A, the largest lymph node. B, the first node receiving direct lymphatic drainage. C, an immune node. Or D, a node near the vena cava. The answer is B. The pearl here is that lymphatic metastasis is a sequential process. By injecting a tracer to identify and biopsy this very first drainage node, we get a direct window into how the tumor is spreading. If that sentinel node is negative, well, the surgeon can confidently spare the patient from the high morbidity of a full radical lymph node dissection.
Shifting gears now to tumor biology and metabolism.
Question 5. What describes the epithelial mesenchymal transition, or EMT? Is it A, muscular encapsulation, B, loss of epithelial traits for a mobile mesenchymal phenotype, C, sarcoma to lipoma transformation, or D, central tumor calcification? The answer here is B. The huge exam pearl focuses on the downregulation of E-cadherin. Normally, epithelial cells are anchored tightly together, but during EMT, tumor cells suppress E-cadherin and transform into this highly mobile, fibroblast-like state. This critical loss of adhesion is exactly what facilitates cell motility, tissue invasion, and ultimately metastasis.
Question 13. What process describes tumor angiogenesis? A, mechanical encapsulation. B, secreting proangiogenic factors like VEGF for new blood vessels. C, transformation into red blood cells. Or D, capillary bed calcification. The answer is B. A tumor can actually only grow about 1 to 2 millimeters on passive diffusion alone. To survive past that tiny threshold, it has to execute an angiogenic switch. Hypoxic cells upregulate HIF1, which drives the secretion of VEGF, essentially forcing the host's body to sprout brand new blood vessels directly into the tumor bed.
Question 15. What metabolic shift characterizes the Warburg effect? A, preference for fatty acid oxidation. B, high-rate aerobic glycolysis over oxidative phosphorylation. C. Shutdown of glucose transport, or D. Direct conversion of amino acids? The correct answer is B. Now, the evolutionary trade-off here is fascinating. Glycolysis produces way less ATP than oxidative phosphorylation, but it happens incredibly fast. And more importantly, it rapidly generates crucial carbon skeleton intermediates. The tumor absolutely needs these building blocks so it can synthesize the proteins and lipids required for constant, rapid cell division.
So just to lock that concept in visually, aerobic glycolysis, or the Warburg effect, is the preferential conversion of glucose to lactate by proliferating cancer cells, even when there is abundant oxygen present. Question 2. What defines oncogenic addiction? A. Absolute reliance on a single hyperactive oncogenic pathway. B. Physical dependency on chemotherapy. C. Accelerated growth in high glucose. Or D. Migration along peripheral nerves. The answer is A. And this concept reveals a massive therapeutic vulnerability. Even though a tumor might contain numerous genetic alterations, it might be entirely functionally dependent on just one dominant driver, like BCR-ABL, for example. If we hit that single dependency with targeted pharmacological inhibition, the cancer cells just undergo massive apoptosis.
Question 8. Which classic tumor suppressor gene is implicated in over half of all malignancies? Is it A. KRS, B. YC, C. TP53, or D. HER2/NU? The answer is C. TP53 encodes the p53 protein enthusiastically known across oncology as the guardian of the genome. Its main job is to halt cell cycle progression so it can repair damaged DNA, or it just forces apoptosis if the damage can't be fixed. We contrast this highly regulated tumor suppressor with options A, B, and D, which are hyperactive proto-oncogenes that just push unregulated proliferation.
Next up, let's explore hereditary cancer syndromes.
Question 9. What is required in Knudson's two-hit hypothesis for a germline carrier to develop cancer? A. Inheriting two mutated copies from one parent. B. Single allele causes cancer at birth. C. Second wild-type allele undergoes somatic mutation. Or D. Entire deletion of proto-oncogenes. The answer is C. The critical concept we're talking about here is loss of heterozygosity at the cellular level. The patient inherits one mutated copy in every single cell. That's the first hit. But a phenotypic malignancy only forms when a specific target cell undergoes an acquired somatic mutation that inactivates that remaining, functional, second allele.
Question 10. Which gene mutation drives Gardner syndrome, which is a FAP variant? A. BRCA1, B. APC or adenomatous polyposis coli, C. RET, or D. P10? The answer is B. Normally, the APC protein targets beta-catenin for destruction, but when the APC gene is lost due to this mutation, beta-catenin accumulates completely unregulated. It translocates straight to the nucleus and drives massive cellular growth. Clinically, this presents with desmoid tumors and highly aggressive early colon cancer.
Question 11. Life-Frumonini syndrome is caused by a germline mutation in which gene? A. MLH1, B. TP53, C. VHL, or D. STK11? The answer is B. Remember, p53 is the guardian of the genome. A germline mutation here globally removes that absolutely vital DNA repair checkpoint. This is just devastating because these patients rapidly accumulate mutations that predispose them to a really broad, severe spectrum of early-onset sarcomas, breast cancers, and brain tumors. Question 14. Multiple endocrine neoplasia type 2, or MEN2, targets an activating mutation in which proto-oncogene? Is it A. RET, B. BRAF, C. MET, or D. PTCH1? The answer is A. The absolute exam key here is that an activating mutation in the RET proto-oncogene drives medullary thyroid carcinoma with a penetrance that approaches 100%. Because of that, identifying a positive RET mutation is an absolute clinical indication for a prophylactic total thyroidectomy during childhood. Let's rapidly review this high-yield matrix just to lock it in. Gardner syndrome is driven by the APC gene, presenting with desmoid tumors and early colon cancer. Life-Frumonini is driven by a TP53 mutation, resulting in those early sarcomas and breast or brain tumors. And MEN2 targets the RET gene, presenting with medullary thyroid cancer and pheochromocytoma.
Finally, let's jump into immunology and targeted therapy.
Question 16. An ER-positive, PR-positive, HER2-negative breast cancer profile classifies as which intrinsic subtype? A. HER2-enriched, B. Basal-like or triple-negative, C. Luminal A, or D. Squamous intrinsic metaplastic? The answer is C. Luminal A is hormone receptor positive and HER2 negative. The major takeaway here is that this specific intrinsic subtype actually carries a highly favorable prognosis. Why? Because the tumor responds so powerfully to targeted endocrine deprivation therapies, like aromatase inhibitors or tamoxifen.
Question 12. What is the primary mechanism of PD-1/PD-L1 immune checkpoint inhibitors? A. Direct cell lysis. B. Blocking the off signal to reactivate cytotoxic T cells. C. Bone marrow suppression. Or D. Artificial growth hormones. The answer is B. You know, tumors survive by exploiting our very own immune system. They express PD-L1 to hit the inhibitory brakes on our T cells. But these breakthrough monoclonal antibodies block that exact interaction. They literally remove the tumor's immunological brakes, aggressively unleashing the patient's own cytotoxic T cells to recognize and destroy the malignancy.
So, as we continue to decode the genetic and immunological secrets of tumor biology, it really makes you wonder, what uncharted pathway will become the ultimate target in the future of cancer care?